Welcome back, everyone. Ready for another deep dive. Today, we're going to explore a fascinating study that links a specific part of a dog's brain to epileptic seizures.
That's intriguing.
It is. Think of the brain as a complex city with different neighborhoods, right? Well, this research focuses on one particular neighborhood.
I like that analogy.
Yeah, it's called the cingulate gyrus, and it seems particularly important in dogs with epilepsy. Now, the source material is pretty technical.
Oh, I bet.
But don't worry, we're going to break it down for you, make it understandable, and hopefully pretty interesting too.
I'm all ears.
So let's set the stage first. We know that idiopathic epilepsy is a pretty common problem in dogs, right? Like 0.6 to 0.75% prevalence.
Yeah, that's a significant number. And a lot of these cases involve what we call generalized tonic-clonic seizures.
Right, those are the ones where the dog loses consciousness, their
Yeah.
body stiffens up, and they might even have those jerking movements.
Exactly. Similar to certain types of epilepsy we see in humans, too.
It makes you wonder if there's something structurally different in the brains of dogs that have this type of epilepsy.
Well, that's what this study was trying to figure out. They focused specifically on the cingulate gyrus.
Okay, so remind us again, what exactly is the cingulate gyrus?
So remember that city analogy? Well, the cingulate gyrus is kind of like a central hub, Grand Central Station.
Kinda.
It's located in the limbic system, which, as you know, is responsible for emotions, memory, and learning. And the cool thing is, it's got all these connections to other important parts of the brain, like the parahippocampal gyrus,
Wow.
the neocortex, the thalamus, you name it.
So if this Grand Central Station isn't working properly, could that create chaos through the whole city?
Exactly. That's what the researchers were thinking. They wanted to know if structural differences in the cingulate gyrus could be playing a role in seizures.
Interesting. So how do they actually go about studying this?
Well, they took two groups of dogs. One group had 32 dogs with what we call idiopathic epilepsy, meaning we don't know the exact cause of their seizures. Okay.
And the other group was a control group of 13 healthy dogs.
Makes sense. Got to compare them to something.
Right. And they put all the dogs through both MRI and EEG.
Hold on. Quick refresher for our listeners. MRI is magnetic resonance imaging. It gives us detailed pictures of the brain.
Yep.
And EEG is electroencephalography, which measures the electrical activity, or those brain waves.
Exactly. And using these tools, they zoomed in on the cingulate gyrus,
Okay.
carefully measuring its volume with some pretty fancy imaging techniques.
So what did they find? Was there a difference between the epileptic and healthy dogs?
There was, and this is where things get interesting. They found that the cingulate gyrus on the same side of the brain as the epileptic activity, which they call the GCC,
You see.
yeah, was significantly smaller in the dogs with epilepsy compared to the healthy dogs.
Wait, so only one side of the cingulate gyrus was smaller?
Yeah, the other side was totally normal. No significant difference in size compared to the healthy dogs. Okay, now that's weird. So one side of this crucial brain hub is smaller and the other side's normal. Does that mean the seizures are causing the cingulate gyrus to shrink?
Well, that's the big question, isn't it? It's kind of like a chicken or egg thing. We don't know for sure if the smaller cingulate gyrus is the cause of the epilepsy or the result of those repeated seizures.
A brain mystery. We've got a smaller cingulate gyrus on the side of the brain where seizures are happening, but we don't know if the smaller size is causing the seizures or if the seizures are causing the shrinkage. Where do we go from here?
Well, the researchers did find some interesting details that might give us clues. For instance, there was this one dog with what we call drug-resistant epilepsy, meaning medication wasn't really helping control the seizures.
Oh.
And guess what? This dog had the biggest difference in size between the two sides of the cingulate gyrus.
Really? So the dog who wasn't responding to medication had the most shrinkage in the cingulate gyrus?
That's
I
concern. It definitely is. It makes you wonder if the severity of that size difference could be a predictor of how well a dog responds to treatment, right?
Yeah, that's a thought. And what about those dogs that didn't fit the pattern at all? The study mentioned a couple where their GCC was actually larger than the other side. What's going on there?
Yeah, those outliers are fascinating. The researchers think it might have something to do with the timing of the MRI.
Timing.
Yeah, like how soon after a seizure they had the scan. Maybe those periictal changes you mentioned earlier were playing a role.
Periictal changes. Can you break that down for me?
Sure. Think of it like ripples in a pond. When a seizure happens, it can cause these temporary changes in the brain.
I see.
And those changes might still be visible on an MRI even after the seizure itself is stopped.
So it's possible those dogs with the larger GCC were scanned during one of those ripples,
Yeah.
and that's why their results look different.
Exactly. It just shows how complex the brain is and how much we still have to learn about these temporary changes.
For sure. You keep saying more research is needed, and I totally get it.
Hmm.
But what kind of research would actually help us solve this chicken or egg puzzle?
Well, one approach would be to do what we call prospective studies, where we follow a group of dogs over time, starting before they develop epilepsy.
It's like a long-term brain diary.
Exactly. We could track changes in the cingulate gyrus as these dogs progress and maybe get a clearer picture of whether the structural differences come first
Right.
or if they happen as a result of the seizures.
That would be amazing. And in addition to those long-term studies, maybe genetic analysis could help too, right?
Absolutely. We know some breeds are more predisposed to epilepsy than others.
Yeah, like beagles and German shepherds.
Right. So if we could identify specific genes linked to both epilepsy and cingulate gyrus development.
That would be huge. Imagine being able to identify dogs at risk for epilepsy early on and maybe even intervene to prevent those seizures from happening in the first place.
That's a really exciting possibility.
Okay, so we've talked about the what of this study. A smaller cingulate gyrus, maybe because of seizures, maybe not.
Yep.
But help me understand the why. Why should dog owners care about the size of their dog's cingulate gyrus?
Well, this research is important because it changes how we think about epilepsy. It suggests we need to look beyond just the seizures themselves and consider the possibility that structural differences in the brain are playing a role.
So it's not just about managing the seizures, it's about understanding what might be contributing to them in the first place.
Exactly. And this has big implications for how we might diagnose and treat epilepsy in the future. Imagine if vets could use advanced imaging to spot these structural differences early on.
Wow.
It could help them predict which dogs might develop more severe forms of epilepsy or which ones might not respond well to certain medications.
It's like personalized medicine for dogs with epilepsy.
Exactly. And if we can really pinpoint how the cingulate gyrus is involved in seizures, then we can start developing more targeted therapies. Maybe find ways to protect the cingulate gyrus or even strengthen its connections to other parts of the brain, potentially reducing the severity or frequency of those seizures.
Now that is incredibly hopeful. But before we get too carried away, let's acknowledge some limitations of this study.
Of course. One big one is that it was what we call a retrospective study,
Meaning,
they were looking back at data that had already been collected.
okay,
Ideally, we'd want to see these findings replicated in a prospective study where we can control for more variables.
makes sense. And there's still that chicken or the egg question.
Yep, that's always going to be there.
Did the smaller cingulate gyrus come first? Or was it a result of the seizures? We still don't have a definitive answer.
We don't, but that's the beauty of science, right? It's all about asking questions, exploring possibilities, and refining our understanding.
Speaking of exploring possibilities, let's talk about the human-animal connection for a moment. Do these findings in dogs tell us anything about epilepsy in humans?
They might. Canine and human epilepsy are actually pretty similar. A lot of the same seizure types occur in both species, and we even use some of the same medications for treatment.
Interesting. So if a smaller cingulate gyrus is linked to epilepsy in dogs, it's possible that similar structural differences could be at play in humans too.
It's definitely a possibility worth investigating.
And that's the amazing thing about research like this.
Absolutely.
Discoveries in one species can help us understand and potentially treat conditions in another.
It's a reminder that we're all connected in this web of life, and discoveries in one area can have huge implications for others.
Beautifully said. It's inspiring to think about the potential ripple effects of this research,
I agree.
extending far beyond just one species.
Exactly. And it highlights how important it is for veterinary and human medicine to work together.
Collaborate.
Exactly. By sharing knowledge and resources, we can make faster progress in both fields and improve the lives of both animals and humans.
Couldn't agree more. So let's recap. What are 3 big takeaways for our listeners today?
Okay, 3 things. First, the structure of the brain matters. We're starting to see that epilepsy isn't just about those electrical misfires.
Right.
There could be underlying structural differences that make some dogs more prone to seizures.
Okay, number 2.
The cingulate gyrus, that central hub in the brain, seems to be a key player in this whole puzzle.
And number 3.
This research could change how we diagnose and treat epilepsy in dogs, and maybe even in humans. We're on the verge of some pretty exciting discoveries.
We've covered so much today, and my brain is buzzing with new ideas and questions.
Mine too. That's what makes this field so fascinating. Always more to learn and explore.
So true. And speaking of exploring, let's dive into some of the specifics of this study. They use a really cool technique called semi-automated volumetry to measure the cingulate gyrus. What is that exactly? Sounds pretty high-tech.
Oh, it is. Remember how we talked about measuring the volume of the cingulate gyrus? Well, in the past, researchers had to do that manually, tracing the outlines of brain structures on MRI scans by hand.
Oh, wow. Sounds incredibly tedious.
It was, and prone to human error, you know? But this study used semi-automated volumetry, which basically uses computer software to help with the tracing.
Ah, so it's like having a computer assistant with, like, superhuman precision.
That's a great way to put it. This technology is a game changer for veterinary neurology.
How so?
It lets us study those subtle structural differences in the brain with a level of detail we couldn't get before.
And that level of detail is crucial for research like this, right? Where we're trying to understand the subtle but potentially important differences between the brains of dogs with epilepsy and healthy dogs.
Exactly. And it's not just about accuracy either. It also makes research more efficient. Researchers can spend less time tracing brain structures and more time analyzing the data.
Sounds like a win-win, especially for the dogs.
Yeah.
So besides this cool new imaging tech, what else stood out to you about the study?
Well, one thing that really struck me was how this research could change how we approach treatment for epilepsy. Right now, we mainly focus on managing those seizures with medication. But what if we could actually address those underlying structural differences in the brain?
You mean going beyond just suppressing symptoms and actually targeting the root cause?
Exactly. If the cingulate gyrus is so important in epilepsy, maybe we can find ways to protect it, or even strengthen its connections to other parts of the brain.
Wow, that's mind-blowing. It's like instead of just fighting fires, we're focusing on city planning, making sure the infrastructure is strong and resilient.
Love that analogy. And it opens up a whole new world of possibilities for treatment and prevention. Imagine if we could identify dogs at risk for epilepsy early on based on those subtle structural differences in their brains.
We could potentially intervene before seizures even start.
Exactly.
That would be amazing. We've talked about the potential benefits for dogs, but you mentioned earlier that this research could also have implications for human epilepsy. Can you tell us more about that?
Sure. Canine and human epilepsy have a lot in common, actually. Many of the same seizure types occur in both species, and some of the medications we use are the same. So if a smaller cingulate gyrus is linked to epilepsy in dogs, it makes sense to look for similar structural differences in humans, too.
It's like having a parallel universe of research, where discoveries in one species can help us understand the mysteries of another.
That's a beautiful way to put it. And it just highlights how important collaboration between veterinary and human medicine is. Sharing knowledge and resources means we can make faster progress in both fields and ultimately improve the lives of both animals and humans.
This study is a perfect example of that.
Absolutely.
Well said. It's inspiring to think about the ripple effects of this research. So as we wrap up, what's the one message you want listeners to take away?
I think the biggest takeaway is that we're entering a new era of understanding and treating epilepsy.
Ooh, I like that.
This research has opened up a whole new avenue of exploration, and I'm excited to see where it leads us.
It's like we've just peeped through the doorway into a vast, uncharted territory.
And the possibilities seem endless.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.
Always. It really is concerning. Makes you think, huh? And to make things even more interesting, there were a couple of dogs in the study that didn't fit the pattern at all.
What do you mean?
Their GCC, remember the cingulate gyrus on the side with the epileptic activity? It's actually larger than the other side.
No kidding.
Yeah.
Larger. So the opposite of what they expected.
Exactly. It's like, what's going on there? And the researchers think, well, it might have to do with when they did the MRI, like how soon after a seizure.
Timing could be a factor.
Yeah, exactly.
Huh.
Maybe those periictal changes we talked about earlier were messing things up a bit.
Periictal changes. You have to remind me what those are.
Oh, right. Think of it like, when you throw a rock in a pond and it makes those ripples.
Okay, yeah.
Well, a seizure can do that to the brain, kind of cause these temporary changes.
Interesting.
And those changes, they might still show up on an MRI even after the seizure's over.
So maybe those dogs with the larger GCE, they were scanned during one of those ripples. And that's why their results look different.
That's what the researchers are thinking. It really shows how complicated the brain is and how much we still don't know about all those little changes that happen.
Absolutely. You keep saying we need more research, and I get it. But what kind of research could actually solve this whole chicken or egg dilemma?
Well, one way would be to do these studies we call prospective studies.
Perspective, meaning.
Meaning we follow a group of dogs over time, but we start before they have any epilepsy.
So like a long-term brain diary.
Exactly. We could keep track of any changes in the cingulate gyrus and see if those differences show up before the seizures start.
Ooh, that would be amazing. And what about genetics? Could that help too?
Definitely. We know some breeds are more likely to get epilepsy than others.
Like beagles and German shepherds.
Right. So if we could find specific genes that are linked to both epilepsy and the way the cingulate gyrus develops.
Wow, that would be a huge breakthrough. Imagine being able to identify those at-risk dogs early on and maybe even do something to prevent the seizures altogether.
Wouldn't that be incredible? It's definitely a possibility worth exploring.
Okay, so we've talked about what this study found. The smaller cingulate gyrus, maybe because of seizures, maybe not. But help me understand why this matters. Why should dog owners care about the size of their dog's cingulate gyrus?
This research is a game changer because it changes how we think about epilepsy in dogs.
How so?
It suggests that we need to look beyond just the seizures themselves and consider that, hey, maybe there are some structural differences in the brain that are playing a role.
So it's not just about managing the seizures, it's about understanding what might be causing them in the first place.
Exactly. And this could totally change how we diagnose and treat epilepsy in the future. What if vets could use these really advanced imaging techniques to see those structural differences early on?
That would be amazing.
Right. It could help them figure out which dogs might have more severe epilepsy or maybe which ones wouldn't respond as well to certain medications.
It's like personalized medicine for dogs with epilepsy.
Exactly. And if we can figure out exactly how the cingulate gyrus is involved in those seizures,
Then what?
then we can start developing treatments that are more targeted.
Makes sense.
Maybe even find ways to protect the cingulate gyrus or make those connections to other parts of the brain stronger. Potentially reduce how bad the seizures are or how often they happen.
Now that is some seriously hopeful stuff. But got to be realistic here. Let's talk about some limitations of the study, right?
Of course. The biggest one is that this was what we call a retrospective study.
Meaning?
Meaning they looked back at data that had already been collected, from cases that already happened.
Okay.
Ideally, it would be great to see these same findings in a prospective study where we could control more of the factors involved.
Right. And of course, there's still that whole chicken or the egg question.
Yep. That one's always going to be there.
Did the smaller cingulate gyrus come first? Or did the seizures cause it to shrink? We still don't have a for sure answer.
Not yet, but that's what makes science so cool, right? It's all about asking questions, exploring all the possibilities, and getting a better understanding of how things work.
Speaking of exploring possibilities,
Yeah.
let's talk about the human-animal connection for a minute. Do these findings in dogs tell us anything about epilepsy in humans?
They could. Canine and human epilepsy are surprisingly similar.
Oh, really?
Yeah. A lot of the same types of seizures happen in both, and we even use some of the same meds to treat them. So if a smaller cingulate gyrus is linked to epilepsy in dogs, it makes sense to see if the same thing's going on in humans.
Yeah, totally.
Yeah.
It's like having this parallel universe of research where what we learn in one species can shed light on mysteries in another.
That's a great way to put it. And it shows how important it is for vets and doctors, human doctors, to work together.
Elaborate.
Exactly. Sharing what we know, pooling resources, that means faster progress in both fields, which means better lives for everyone, humans and animals.
This study's a perfect example of that.
Absolutely.
Well said. It's so inspiring to think about how far-reaching this research could be.
I agree.
So as we wrap up this deep dive, what's the one big takeaway you want our listeners to remember?
I think the biggest thing is that we're on the verge of a whole new era in how we understand and treat epilepsy.
Ooh, I like that.
This research opens up so many new avenues to explore, and I'm really excited to see what comes next,
It's like we've just opened the door to this huge, uncharted territory.
Exactly.
And the possibilities are endless.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.